Hi, this is Jeff Hoffman. Let's move on to genetics. Just like most things in medicine, genetics has a set of terminology that you should be familiar with. Let's get right into it. Codominance occurs when neither of two alleles are dominant or recessive, but both contribute to the phenotype in a heterozygote. An example would be blood types. This means that in patients who are heterozygous or have one of each type of allele, A and B, each allele has an effect. Variable expressivity is when individuals who have the same genotype have a different phenotype. For example, two individuals who both have the same mutation in NF1 can have different presentations of neurofibromatosis. The patient on the left has some small cutaneous neurofibromas and a café au lait spot, and the patient on the right has lots of very large neurofibromas but no café au lait spots. Incomplete penetrance is when not all individuals with a certain mutant genotype show any type of phenotype. This is different than variable expressivity, since in the case of variable expressivity, all patients show some type of phenotype. It just varies from one to the next. An example of incomplete penetrance is the BRCA1 mutation, which can cause breast cancer in some people, but have no effect in others. By the way, do you know what the BRCA protein does? It's used to repair double-strand breaks in DNA. In pleiotropy, a genotype can cause multiple different phenotypic effects. So you might say that a gene has pleiotropic effects. For example, what are some of the effects of phenylalanine hydroxylase deficiency, which causes phenylketonuria? These patients can have such symptoms as intellectual disability, lighter colored skin, hair, and eyes, and a musty body odor. This might sound kind of similar to variable expressivity, but variable expressivity is the occurrence of different phenotypes in different people, whereas pleiotropy is a variety of different effects in the same person. Anticipation is the process by which the severity of a disease worsens or the age of onset of the disease is earlier in succeeding generations. In other words, if your maternal grandfather has a mild disease, and then your mother has a moderate version of that disease, and you have the most severe version. An example of this is Huntington disease, which we'll talk about more later. Loss of heterozygosity is something we usually talk about in the context of cancer, or more specifically, tumor suppressor genes. So let's say we have these two chromosomes in blue, and the red line represents a gene, specifically a tumor suppressor gene, like maybe p53. If one of these genes is knocked out, or has a mutation that makes it not functional, we have a heterozygous state, and the remaining copy on the chromosome is sufficient to prevent tumors. But if both copies are defective, then we have lost heterozygosity and are no longer able to prevent tumors. This is also referred to as the two-hit rule. So what do you think about oncogenes? Does this rule apply to them also? The answer would be no, because even one single mutation in an oncogene is sufficient to cause cancer. You don't need the other copy to be mutated as well. Remember, a tumor suppressor gene is something that shuts down the cell cycle, and an oncogene is something that stimulates the cell cycle. A dominant negative mutation is one that prevents normal function of the gene but has a dominant effect. So even if only one allele has a dominant negative mutation, its protein product will prevent the protein product of the wild-type allele from functioning. For example, let's say you have a transcription factor, and a transcription factor needs to first bind to a specific DNA sequence, usually in response to another stimulus, and then initiate transcription of the nearby gene. Now let's say that one of your two genes that code for this transcription factor is mutated in such a way that it will bind too tightly to that DNA sequence, but it won't initiate transcription. Now it will bind all the time, so the normal healthy copy will never get in there and will never transcribe the gene. Linkage disequilibrium is a tricky one, so I'm going to bring back my chromosome model to make it easier to understand. Now normally we have 23 pairs of homologous chromosomes, but let's pretend we just have 3 to make this simpler. I'm showing some genes here as red lines. Now remember, during meiosis, we're going to have these chromosomes segregate independently, and we'll end up with one of each in each gamete. I'll give these genes names. So let's say this is A, B, C, D, and E. And in the other gamete, I'll use lowercase letters. So again, we have little a, b, c, d, and e. Now before these separated, since chromosomes segregate independently, gene big A had a 50-50 chance of ending up in either the gamete with the big D or the little d. So what were the odds of the big A ending up with the big C? Now this is kind of a tricky one, but it's actually also about 50%, because they're so far apart on the chromosome that a recombination will happen a few times between them as the cell prepares for meiosis. Now, recombination is something we don't really get into in first aid, as it's not very high yield for the step 1, but just think of it as homologous chromosomes, just like the two blue ones, swapping homologous regions. Do you know what it's called if two non-homologous chromosomes, like the blue and the green one, swap chunks of DNA? That's called translocation, and I'll talk about that more later. Anyway, what do you think the odds are that the big A and the big B will stay together? Now these have a very high chance of ending up together since they're so close in the chromosome that there's a very low chance that recombination will happen between them. Now here's where linkage disequilibrium comes in. Let's say you only have little a's and little b's in a population, but suddenly in one person you see two new mutations, big A and big B, which are dominant, and big A produces, let's say, green eyes, and big B produces brown hair. And these are both new mutations, so nobody else in the population has green eyes or brown hair yet. Now for many generations after this, almost everyone who has green eyes will also have brown hair. So to put this mathematically, after a few generations, you might see an equal distribution of big A's and little a's, and big B's and little b's, so 50% of each. Now by normal Mendelian genetics, among all the chromosomes in the population, you would expect to have about 25% of each combination of B's and A's. However, since big A and big B are in linkage disequilibrium in the population, you might actually see a lot of chromosomes with big A's and big B's, and a lot with little a's and little b's, but hardly any with a combination of the two. So mosaicism is when some cells in the body have a different genetic makeup than others. This can only be caused by events that occur after fertilization, since anything that happens before fertilization will have the same effect on all cells in the body. Mosaicism can be caused by mutations that occur during embryogenesis. For example, if a patient would have an AB blood type, but a deletion of the A gene occurs in the cell that gives rise to half of the hematopoietic system, then half of the patient's blood cells will be AB, and the other type will just be A. A second way mosaicism can be caused is if two zygotes form, which would have ended up as dizygotic twins, except they fused together very early in embryogenesis. Now half of the cells will have the genotype that one twin would have had, and the other half of the cells have the genotype of the other twin. One last example is germline mosaic, which is when a mutation occurs that only affects cells that will give rise to the germline. This will have no effect on that individual, but will affect their offspring. The offspring will not have mosaicism since they will be derived entirely from a single cell, but they can have a mutation that was not present in their parent's somatic cells, causing a disease that the parent did not have. I also want to point out that random X chromosome inactivation in females, or lionization, is not an example of mosaicism, since all cells still have the same genetic material. The only differences between cells are epigenetic modifications that shut off one chromosome, which doesn't count as mosaicism because even in other chromosomes, different cell types have epigenetic differences, such as a neuron compared to a hematopoietic stem cell. An important clinical point about mosaicism is that some genetic diseases can only exist in a mosaic individual, since the genetic mutation is so severe that if it affected all cells it would be lethal. An example is McCune-Albright syndrome, which is caused by mutation in the GS-alpha gene, which is part of the G protein signaling cascade. If this gene were mutated in all cells, it would be embryonic lethal, but individuals who are mosaic for the mutation can survive. So how does this present? Well, first of all, you'll see these unilateral café au lait spots. They can present on both sides of the body, but they don't cross the midline. Second, these patients have polyostotic fibrous dysplasia, which is when parts of some bones are replaced by fibrous tissue, and as you can see here, the most common locations are the base of the skull and the femur. This bone pathology also results in facial deformities. Locus heterogeneity is when mutations at different loci, or in different genes, produce the same phenotype. This can occur if two genes have the same function, or if they act in a sequence in a pathway. For example, Marfan syndrome, MEN2B, and homocystinuria can all result in a marfanoid habitus, which means the patient is tall and thin with long fingers and joint hypermobility. There are also several genetic mutations that can cause albinism, such as in genes responsible for the production of melanin and genes involved in the transport of melanin from melanocytes to keratinocytes. Allelic heterogeneity is kind of an obvious principle, and basically it's just that if you mutate a gene in one place and it produces a certain phenotype, then mutating a different part of the same gene can cause the same phenotype. This is probably true more often than not, so it seems unnecessary to give one example. But in the book we have listed beta thalassemia, which can be caused by different mutations in the beta-globin gene. Heteroplasmy is the presence of two different versions of mitochondrial DNA, both existing in the same cell. Since you only inherit mitochondrial DNA from one parent, your mother, this occurs due to new mutations rather than the inheritance of different genotypes. A mutation can propagate, resulting in some mitochondria having the mutated form and others having the normal form, and both can be passed on to new cells after division, which is especially important if it happens in either an egg or soon after fertilization, since more cells can potentially inherit the mutant DNA. This can result in variable expression of an inherited mitochondrial disease, depending on how many cells get how much of the mutant DNA. Our last term is uniparental disomy, which can happen when an offspring receives two copies of a chromosome from one parent and no copies from the other parent. There are two types, heterodisomy and isodisomy. Heterodisomy is caused by non-disjunction in meiosis I, and isodisomy is caused by non-disjunction in meiosis II. So what's the difference in the final gamete? I'll label these here to walk you through it, and we'll do a little bit of review of meiosis. You have two homologous chromosomes which are genetically different because one of them is from your mom and one's from your dad. So I'm labeling these different homologous chromosomes as big B and little b, and those each replicate into two sister chromatids, so you have two big B's and two little b's. Normally in meiosis I, the genetically identical sister chromatids stick together. So you'll have the two big B's in one cell and the two little b's in the other cell. And then in meiosis II, they separate into one per gamete. So if non-disjunction is in meiosis I, you'll end up with one big B and one little b in each gamete. If meiosis II is the culprit, then some gametes will be normal, but the other one will have two little b's, or no B chromosomes at all. This should make sense with the terms here, with hetero meaning different, like you have one big B and one little b in each gamete, which are two different chromosomes, and iso meaning the same, so here you end up with two of the same chromosomes in a gamete. As a side note, isodisomy can also be caused by duplication of one chromosome and loss of the other. So what's the effect of uniparental disomy? Well, remember, the other side of uniparental disomy is that the gamete received from the other parent is missing this chromosome. So the person will be euploid, meaning they have the correct number of each chromosome, so they often have no disease trait and have normal fertility. But between a heterodisomy and isodisomy, which do you think is more likely to cause disease? This would be isodisomy, because if the parent was a carrier for a rare autosomal recessive genetic disease, their offspring will end up with two affected copies and thus have the disease. This can sometimes look like in printing, which we'll talk about soon, since you're only receiving genes that can be expressed from one parent,